This study was designed by Data Acquisition Working Group of the NCI Quantitative Imaging Network to characterize and correct for major sources of systematic errors in parametric maps of apparent diffusion coefficient, ADC. Such errors deteriorate intersystem reproducibility of quantitative imaging metrics in multi-center clinical oncology trials. This project was carried out by research teams from 7 cancer imaging centers that utilize 14 clinical MRI platforms representing 10 distinct system models by 3 vendors at 2 magnetic fields. ADC is a quantitative measure of mean diffusivity of water which is modulated by the cellularity of the imaged tissue that in its turn can be altered by disease or treatment. The ADC is derived from 2 or more diffusion-weighted measurements assuming mono exponential signal decay with increasing diffusion weighting, quantified by B-value. B-value is determined by the strength and timing of applied diffusion gradients. The imperfections of the gradient channel hardware may cause significant non-uniform deviations in diffusion weighting from its nominal value. Such systematic deviations introduce bias errors in tissue ADC maps that broaden and shift ROI histograms, thereby lowering their diagnostic power. To evaluate the extent of the systematic bias across diverse and commonly used clinical MRI platforms, our research team implemented uniform DWI acquisition protocol using temperature-controlled isotropic ice water phantom of known ADC value placed on the platform with controlled elevation. Individual gradient channels were characterized independently by recording ADC along primary magnet axis at 3 offset, right-left, and superior-inferior. The observed offset-dependent bias was quantitatively described using quadratic fit coefficients related to shift, skew, and curvature. The bias fit coefficients correlated strongly to specific characteristics of the gradient systems, such as gradient non-linearity, GNL to curvature and all channel shift, imaging gradient cross terms to imaging channel shift, background shim to bias skew, and eddy currents to ROI uncertainty and goodness of fit. To identify dominant bias sources across platforms and define appropriate correction strategies, a comprehensive model was developed for the DWI sequence variants utilized by different vendors, single spin echo and double spin echo. The developed model included observed gradient imperfections from GNL, shim, imaging, and EC. By this model, the ratio of bias to nominal B-value directly predicted behavior for the measured fractional ADC bias ratio to known value. The fractional ADC bias derived from channel average fit as a function of right, left, and SI offset for all measured systems confirms significant system-dependent and spatially non-uniform deviation from nominal diffusion weighting, ranging from positive 25% RL to negative 55% SI. The prevailing quadratic curvature of the observed bias was consistent with GNL origin. In fact, GNL correction by itself removed more than 80% of total ADC bias error for all systems. For the offsets greater than 5 cm, the observed bias resulted in average 20% variability across platforms. The minor errors due to constant shift and relative skew of the ADC traces, as well as fit uncertainty within ROI, were below 5%. The evaluation of channel-specific GNL and skew as a function of field strength and sequence variant revealed dependence of GNL bias on gradient system design, channel geometry, and nominal independence of sequence variant. The nature of the dominant bias source in excess of 10% for most systems was clearly different from that of relatively minor shim-related skew bias, which was independent of gradient channel design and was reduced for DSE acquisition sequence. The observed effect of DWI sequence variant on B-value bias was consistent with model prediction for the corresponding gradient waveforms. Both experimental top and simulation bottom confirmed immunity of GNL bias to DWI sequence specifics and ability of DSE sequence variant to mitigate minor bias sources due to imperfect shim and imaging cross terms. In summary, the implemented protocol allowed empiric description of DW bias for individual gradient channels, regardless of vendor platform, and determination of the dominant sources of ADC error for multi-center trials. The results confirmed gradient nonlinearity as a significant source of bias in ADC maps of center, not currently corrected on clinical scanners. Additional minor sources of diffusion weighting bias due to shame and imaging gradients could be mitigated by twice refocused spin echo acquisition. Correction of GNL bias significantly improves ADC accuracy and reduces variability in multicenter studies. Note that GNL is deterministic property of gradient system, and corresponding bias in principle can be corrected online on the MRI systems similar to geometric distortion correction.